I'm Vaughn Mancha. I'm a physician. I actually practice just down the road here at American Family Care. I'm boarded in family practice. And since medical school, we're actually in college, I've always felt like, the issues that I was taught about evolution just didn't make sense. Things were just too complex for things like that to happen. And so I've always kind of been following on the internet, reading vociferously different articles about evolution to see if things were going to change. And what I'm going to tell you about tonight is a field of what's called epigenetics that's come out that is blowing the lid off of everything. It's completely changing everything we knew. And not only that, it's also making Darwinism a really moot point at this, and which is kind of fascinating because the scientists themselves are saying, oops, we made a mistake. And Kyle already talked about Ratio Christi and the fact that we've got an organization now, and we're going to be meeting on campus, I would imagine, by the next time. Our next meeting, by the way, we're going to have Steve Biggs, who wrote a book called Beyond a Reasonable Doubt. He's a chemical engineer, and he's lectured to numerous churches and other organizations about the things he's discovered. So the title is going to be Epigenetics, or Does Darwinism Need a Rethink? And that's just not me saying this. It's the scientists themselves saying this. What's really fascinating is this little phrase here in Proverbs. It's to the glory of the God to conceal a matter. And in fact, this whole issue has been concealed really until the last 10 years or so. And as a result of it, the Darwinists and others completely threw in to a certain way where they said it has to be this way. They completely ignored this whole field that is just springing up. And so it's like God gave them enough rope to hang themselves with, because now they're like, uh-oh, we can't change our theory. The theory is locked in to a certain fashion. I'm going to show you how it got locked in with a few big concepts. This is kind of an interesting passage because this passage was actually quoted in a science book talking about epigenetics. It says, I am the Lord your God. I'm a jealous God, visiting the iniquity of the fathers to the children on the third and the fourth generations of those who hate me. This basically says that hatred, anger, can actually be passed through our genes. That was absurd from the way we had learned genetics in the past. But epigenetics actually works 3 to 4 generations. We can actually measure things out. With rats, for instance, under stress, that their great-grandchildren will feel the stress that this particular rat felt and other things like that. So it's actually this, I used to think, isn't that kind of unfair? God is visiting a sin to another generation, when actually it's a warning that because of your sin, the sin of hatred, that anger and angst will be passed on to your great-grandchildren. And this is also why you can see whole societies that are full of anger and everything that just can't break out of it. So this is the article that came out in the journal Nature, which is the journal on biology and evolution. And look what it says. Does evolutionary theory need a rethink? That's an astounding admission to even raise the question. It would be anathema or sacrilegious to even question evolution, but here they are, Journal Nature, and all the people, this is just a year and a half ago. What did they say? They say, does evolutionary theory need a rethink? Yes, urgently. They're not paying attention to certain key processes. And then they go on to talk about Charles Darwin. He conceived of evolution by natural selection without knowing that genes existed. Now, mainstream evolutionary theory has come to focus almost exclusively on genetic inheritance and the process that changes gene frequencies. This is what they study when they study natural selection. Said, yet data is pouring out of adjacent fields that are starting to undermine this narrow stance. Darwinism, a narrow stance, these guys are saying. An alternative version of evolution is beginning to crystallize, in which the processes by which organisms grow and develop are recognized as the cause of evolution. Now, when they say evolution, they're saying change. They're not saying Darwinian evolution with this. What they're saying, basically, is that our interactions with the world or chemicals that we come into contact, other health issues affect our DNA and we transfer it onto our children. But what is Darwin's natural selection? And of course, we have the famous book, The Origin of the Species by Charles Darwin, written around 1850, I want to say. Natural selection was first coined in that book. And basically, Charles Darwin looked at animal husbandry, the interbreeding of animals, and he saw that they could change and do remarkable things with the interbreeding of animals. And he thought, I wonder if in nature this sort of selection occurs. In other words, random breeding under natural selection out in the wild can lead to more fit offspring, and they're the ones that survive, the survival of the fittest. Everyone's heard that. It makes a lot of sense, and in fact, it does work in certain systems like bacteria and fruit flies and some other things. But when it comes to trying to go all the way from a bacteria to a human being, it doesn't go that way. Darwin didn't know what the source of fitness was, genetics. He just assumed that life kind of morphed from one thing to the next. He looked at cells as just kind of like jelly donuts that weren't really all that sophisticated, but one single human cell is more complex than the Airbus factory that we have on the edge of our town. I mean, it's got so much information in there. And in one cell, you have 6 feet of DNA. If you took the 10 trillion cells we have and stretched the DNA end on end, it would go to the sun and back 100 million times, actually, 100 times, excuse me. So we're talking about millions of miles of instruction sets in our DNA, very complex. This gentleman is the name of August Weismann. He was a very famous zoologist that really shaped kind of one of the first mistakes that we really kind of went down. We took Darwin's theory and then we added Weismann's barrier. And what is Weismann's barrier? Weismann said that there's a separation between our germ cells, our gametes, and our bodies, the soma, so that the changes to our bodies do not influence the germ cells and inheritance. Now, many people still believe this today, that for instance, we can have a bad cup of coffee or something and it won't affect our DNA and our genes and our gametes. But that's actually being shown to be completely different. In other words, the Weismann barrier, the separation of our gametes from generation to generation is falling down. And it actually kind of headed us off into a way of thinking that really kind of skewed our thinking on biology completely. It says this idea of true rules out inheritance of acquired characteristics. And this was first proposed by John Baptiste Lamarck. So basically going back to that Bible verse, the hatred going from generation to generation, this is saying that couldn't happen. And it seemed to make sense. I mean, when I was in biology in college, it made complete sense. This is John Baptiste Lamarck. He lived 50 years before Darwin, and he was the popular theory at the time. He believed that our experiences and our environment could influence our germ cells and inheritance. And there were just huge battles over this whole thing. And Weismann really, even though Weismann was a good biologist, he basically wanted to destroy this concept of Lamarckian. It was just like a huge battle back in the day. Adding to this confusion, we have Francis Crick here, who was one of the co-discoverers of the DNA molecule. He and Watson and Crick, you've probably heard of, discovered the DNA molecule, and he came up with the concept of what's called the central dogma. So like a dogma, like a church papal bill that comes down. It's got to be this way. And people are following the central dogma even up till today. The central dogma is basically that you take DNA and then you make RNA from it and then you make protein, and it doesn't go the other way. That's actually true in a sense, but it's not true in the fact that the protein and the RNA are feeding back and causing the DNA to do different things. But this kind of simplified things. So you take Weismann's barrier and Crick, and you come up with roughly what we call today the modern synthesis. These are 2 key things to it. And again, you see the germ lines going from one germ to the next. The sperm and the egg creating a whole new set of sperm and egg, and then the cells break off to be the soma. And if you think about it, our our seed and everything tends to go on kind of indefinitely, but our bodies don't. So in a sense, that kind of makes sense that that works that way. And then you come along and you take the central dogma on the bottom where DNA only goes to DNA and then protein comes off and it can't go back to DNA. And then you kind of fuse those two things together by a gentleman called John Maynard Smith, who's one of the leading fathers of genetics and biology. And we kind of took those two things and just kind of accepted them. It seemed to make sense. And in a lot of ways, there's truth to them, but in a lot of ways, it completely kept us from looking for the real truth. Kyle, is he here? I'm going to get you to click on this YouTube link. I'm going to show you the basic understanding of mutations and biology with this YouTube on the blue link there.
The errors or mutations that occur in the replication of DNA can have significant impacts on individuals and on species. Mutations that occur within body cells can, for example, cause illnesses such as cancer if the mutation programs the cell and all its daughter cells to grow and divide uncontrollably. Such mutations are often caused in humans by radiation and chemicals, such as those found in cigarette smoke, that break strands of DNA. As we will see, the resulting repair work often leaves the DNA with an error. Mutations that occur in sex cells don't affect the individual in which they occur, but rather, if they are carried in a sex cell that unites with another sex cell, they are carried into the next generation. Such mutations have the potential not only to affect the individual that initially inherit them, but in the long term, the species as a whole. That is because an individual who inherits a mutation has a copy of that mutation in every cell of their body, including the ones they will eventually use in the production of sex cells. If the mutation increases the fitness, as judged by natural selection, of the individual and succeeding generations, the mutation will likely become more and more common in the species population. Each and every one of us are largely the result of mutations that, starting with the first piece of DNA in the first living organism, have been successful in increasing the fitness of individuals and species as judged by natural selection. We are, therefore, the products of successful mutations and the adaptations they led to. But history is full of even larger numbers of mutations that were judged by natural selection as unfit for survival. There are three basic types of mutations: point mutations, insertions, and deletions. Occasionally, during replication, in spite of all the proofreading and corrections, a mismatched base pair in one of the chromosomes, as for instance an adenine base pairing with a cytosine base, goes unnoticed, and the cell proceeds with either mitosis or meiosis. Repair enzymes in the daughter cell that received the mismatched pair notice the mismatch and proceed to correct it. However, these enzymes no longer have any way of knowing which strand is an original parental strand or a newly replicated daughter strand. If the enzymes correct the mismatch by replacing the base on the daughter strand, the original base pair will be restored. However, if the enzymes correct the mismatch by replacing the base on the original strand, a base pair different from the original DNA will result, and a point mutation will have occurred. Insertions and deletions occur not due to any errors in replication, but rather from a break or breaks in one of the strands. Often outside sources, such as radiation or chemicals, cause these breaks. Insertions often occur when one strand breaks in a single location and one or both of the freed ends loop out. Repair enzymes fill in the resulting gap with new nucleotides, making the strand that had the break longer than the strand that remained intact. As a result of how the process of replication works, after cellular division, one daughter cell will receive a new longer segment of DNA due to the insertion mutation, while the other will receive an unchanged, mutation-free segment of DNA. Deletion mutations occur if a strand of DNA breaks in two places. The section between the breaks is forced out, and the repair enzymes draw the two remaining strands together without filling in the missing nucleotides. Again, a loop occurs in one of the strands, but in this case, it is the intact strand. Now when cellular division occurs, one daughter will receive a shorter segment of DNA due to the deletion mutation, while the other strand remains unchanged. Having seen how DNA replicates itself and how mutations occur, we now look at how the genetic information contained in an individual's DNA is converted to proteins that make living organisms what they are.
Okay, so that is the theory of evolution as we used to understand it. So basically mutations causes better genes, and and you can see the kids playing soccer, so obviously it's true. They're better soccer players, and the elephants are better elephants and things like that. Well, there's a lot that they didn't discuss, isn't there? I mean, supposedly the mutation goes on to make a better protein. When you really start to do those studies, the whole concept kind of breaks down. But nevertheless, these are actual real things. These genetic mishaps do occur. So we're not saying that they don't occur. It's just that the focus was completely on mutations. I mean, and after all, you've been to see the Fantastic Four. You know, the guy gets mutated by the radioactive waves, and he comes out to be a bigger monster, right? I mean, so mutations obviously works. So that is the.
Can I ask a question?
Sure.
That example was on somatics, not germ cells.
Well, if you noticed that it was talking about the mutation will only get transmitted if it occurs in the germ cell line. So there's that Weismann barrier separation again, that a mutation that occurs to our outer body, our soma, doesn't go to our kids. So how in the world are they going to gain from our experiences or the things that happen to us? Well, and that's again because of the way they look at things. Let's see. So let me back up. So that's kind of the modern synthesis. It takes the central dogma of DNA to RNA to protein. It adds the Weismann barrier, which says the germ cells are separated from the somas, and then it also throws in mutations to say that the genes are changed that way. And again, these things do happen, but they happen on the very small, like bacteria and other things, to cause real natural selection. On the big case, it really doesn't. So we're recalling the central dogma, RNA to DNA to RNA to protein. And our thinking was that, well, if we have a disease process, it must be from a disease-mutated DNA, right? I mean, and we thought that if we could get the human DNA and completely type it out, well, all we got to do is find those mutated DNAs and correct it, and all problems will go away, right? So we launched into the Human Genome Project, which started in 1990 and completed in 2001. And Francis Collins, who is the head of this, is now the head of the NIH. So they typed out 1 billion base pairs, 3 billion base pairs basically, to discover that we have roughly 30,000 genes. And we thought, great, we're now going to figure it out. So we typed it out, and then what happened? Nothing. It was a remarkable silence of research. In fact, I was doing clinical trials for about 13 years, and nothing was coming out about DNA changes at all. Even though we had the human genome out, why weren't we going in there and adding things to change it? Because it wasn't the case. I mean, we knew that there were schizophrenics. For instance, twins, consider twins, identical DNA, right? but one twin would be schizophrenic or have manic depression or something else. How can that be? They have the same exact sequence. Supposedly by the central dogma, they must have a bad piece of DNA, but that's not the case at all. Same thing with diabetics. They have the same genes for insulin as the next person. What are we missing? You know, we completely went off the wrong way. This evolutionary thought that we had us going that particular way. I mean, I went all the way through medical school thinking the same thing. During all this that was going on, there's going to be a test on this, by the way. Okay, these things were happening. Okay, you know, here's Crick's discovered the DNA molecule back here in 53. In 58, the central dogma came along, but then they started discovering these other things, that there was different types of RNA that were doing all kinds of different things. And there's just all kinds of different RNA that was being discovered over that 10-year period that we were pretty much ignoring, basically. And then finally, we came down to this project called the ENCODE project, which after we realized that we weren't hitting any pay dirt, we said, uh-oh, we're going to have to study all that extra stuff that's surrounding the DNA, that's making the DNA work in a certain fashion. So they formed what's called ENCODE. It started right after the Human Genome Project. And the fact that the Human Genome Project wasn't getting anywhere, they said, okay, we'll start looking at different cell types. They studied 150 different cell types. And they discovered that 80% of the human genome was doing a certain thing, while the evolution biology was saying only 2% was active. They were saying, no, there's this other 80% that's also active. There was different protein coding genes. There was different RNA genes. The RNAs were doing things to the DNA. The DNA was doing things to the RNA. The protein was doing things to the RNA. So this was a huge project. It was more than 400 researchers. It was 400 large lab. There's literally thousands of researchers involved in this whole project. And the data they've gotten out, they're still publishing today. I have articles that come to my email just based on ENCODE daily, 20 articles daily. It's not surprising. So here we have, going back to the central dogma, our DNA, RNA, protein, but uh-oh, there's all this other stuff going around that's far more complex. You know, the DNA is wound up in little beads, and we'll show that in a YouTube in a second. The DNA is changed by different factors. The cells are feeding back on everything. There's all these different types of DNA, of RNA that we discovered. It's not just messenger RNA. There's all kinds of different pieces of RNA that's doing different things. We have epigenetics, which basically means above the gene. If you think about it, think of the DNA as kind of the computer, and the epigenetics as being the software that makes the computer do things. So epi, meaning above the gene, causes all these different processes by modifying the DNA, the RNA, and the proteins all at the same time. Now I'm going to show you some bizarre, complex slides. This is just to show you how much more complex it really is. Okay, so we have this going on. All these different mechanisms and feedback mechanisms that are going on, and this, and this, and this. All these are just more and more complexity than that little film that you saw a little while ago where DNA just makes RNA, makes a protein, and then suddenly you're playing better soccer. That's not the way it works. This versus this. This is back to the Weismann-Berry and the central dogma connected together. Very simple approach to things. We thought everything seemed so simple. So, are you with me so far? Okay, let's look at epigenetics in an illustrated fashion because they'll really explain a lot. And we're going to watch both of these clips.
There are over 100,000 skin cells in 1 square centimeter of skin. Each of these cells contain close to 2 meters of DNA, which contain our genetic information. All the genetic material in our body is called the genome. Because our DNA is very long, cells package up their DNA together with proteins known as histones. The DNA and protein complex is known as chromatin. DNA is wrapped around histone proteins, forming repeated units of nucleosomes that look like beads on a string. chromatin is condensed further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is composed of DNA wrapped around a histone octamer that consists of 2 copies of the major types of histones, H2A, H2B, H3, and H4. This organized DNA protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cells will have genes turned on that a muscle cell requires, and will turn off genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes that a liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides, G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG island. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modifications. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
Remember in the first clip, it was saying that cancer is caused by mutation. But in fact, cancer is caused by the genes that fight cancer being turned off. Huge difference. You know, we had been completely buying in, again, you know, so, and this is another clip talking further about epigenes and how it causes inheritance. So basically breaking the Lamarckian, the Weismann barrier.
Epigenetic modification of the epigenome is established during early embryonic development and is maintained during cell division. In this video, we will look at DNA methylation during embryonic development and the influence the environment can have on DNA methylation patterns. During the period of embryonic development, methylation patterns of the germline and somatic cell lineages are established. Here is a pregnant woman, and we will zoom into the uterus, and from here go back in time to when fertilization occurred. Then we will follow the changes in DNA methylation pattern during embryonic development. Initially, the egg that is released during ovulation carries the maternal DNA methylation pattern. The sperm that will fertilize the egg carries the paternal DNA methylation pattern. it is important to draw a graph. Here we have on the y-axis the level of DNA methylation pattern, and it can be increasing or decreasing during early embryonic development. The female egg and male sperm, as I already mentioned, carries its own DNA methylation pattern. During fertilization, a zygote is formed, and the maternal and paternal genome and epigenome are contained in one cell. The paternal epigenome then rapidly undergoes genome-wide active DNA demethylation and remains demethylated following multiple rounds of cell division. The maternal epigenome undergoes gradual, passive demethylation. By the morula stage, the methylation pattern of the embryo is essentially erased. At the blastocyst stage, de novo methylation is initiated. De novo methylation is where new methylation patterns are re-established in the growing embryo. During differentiation, each cell type acquires a unique DNA methylation pattern that is subsequently maintained during each cell division. The initial enzyme responsible for removing DNA methylation patterns from the maternal and paternal genomes are enzymes called TET. Methylation are carried out by DNMT enzymes. DNMT3A and DNMT3B are responsible for de novo DNA methylation of embryonic cells. And DNMT1 is responsible for maintaining the DNA methylation state in somatic cells during each cell division. DNA methylation plays an important role in human health and has been associated with a growing number of diseases, including cancer, imprinting disorders, and repeat instability diseases. Although the DNA methylation patterns are relatively stable, changes in methylation can potentially occur during early development due to environmental influences. For example, the mother's environmental exposure to smoke, alcohol, nutrition, and drugs may lead to random epi-mutations in the developing embryo, and even in the germ cells of the developing embryo. The developmental origins of health and disease hypothesis proposes not only that we are what we eat, but also that we could be what our parents and grandparents ate, and what they have been exposed to. The epigenome, unlike the genome, can also be modified in response to interactions with environmental conditions after birth, and accumulated DNA methylation alterations as we age can lead to diseases such as cancer.
So you see, this is a complete difference than the Weismann barrier, which says that our germ cells are different from our somatic cells. The DNA sequence doesn't change, but how the DNA sequence is used is completely changed. So you can see that when we got into this concept of the central dogma, added the Weismann barrier, add natural selection, and these guys were just chunking out paper left and right and completely ignoring all this stuff. You saw the first film, it didn't talk about any epigenes. It was just a mutation caused everything, with a lot of arm waving, and the kids are playing better soccer, right? I mean, are the walruses going down the beach? It has nothing to do with anything. The behavior of the walruses, for instance, is all epigenetic. How they cry, how they talk to their mothers, all these are epigenetic learned behaviors, and how their genes are expressed is completely different than we were thinking. Eva Jablonka, who's one of the leaders in epigenetics, she was in that first article about does evolution need a rethink. She was one of the key people. She's over in Tel Aviv, and it's kind of like, it's interesting, you almost have to get out of this country to get away from all the talking heads that are telling each other the same stuff. And she's been out there waving her arms saying, guys, you got it wrong for the last 15 years. And she's going to talk about how people had it wrong, basically.
Well, what I'm doing really is theoretical work, and I'm mainly interested in the evolutionary implications of epigenetic inheritance. So the more we learn about epigenetic inheritance, the more we understand how traits are epigenetically transmitted from one generation to the next, the more we can develop substantial models in evolutionary biology about the evolutionary impact of epigenetics. So at the moment, what is happening in evolutionary biology is that people are beginning to consider epigenetic variations and the inheritance of epigenetic variations as a factor in evolution. There is a lot of resistance to this, and the resistance is due to the fact that the epigenetic system has a dual role. It is both a response system, epigenetic control is part of regulation of gene expression, and it is also a memory and inheritance systems. Because of this dual role, induced changes that change epigenetic marks can be transmitted from one generation to the next, at least in theory, and it seems also in practice, because we are seeing a lot of transgenerational effects that we don't exactly know how specific they are, but sometimes they seem pretty specific, and they're inherited sometimes transiently, sometimes for many generations. Now, this kind of thing is resisted because it is felt that it is going against the prevailing view of evolution, where selection, natural selection, is the only factor that is shaping adaptation. If you introduce epigenetic inheritance and the inducible aspects of epigenetic inheritance into evolutionary theory, it looks like Lamarckian inheritance. It looks like you have the inheritance of acquired epigenetic variations, which are or can be sometimes specific response to the environment. Now, a lot of people do not like this. A lot of people regard it, have a kind of knee-jerk response to it. Cannot be true. We have learned in high school that this is not true. Lamarck was wrong. Well, I think that we have to reconsider it. And that is why I think evolutionary theory, evolutionary studies has been lagging behind all other domains in biology in terms of the impact that epigenetic research is having on them. This is changing. I think that what we need, and what we need really badly, in addition, of course, to empirical studies, are good models. And we need the Haldanes, Fishers, and Wrights of epigenetics. We need people who will systematize the knowledge that we have at present and the theoretical implications of variable transmissibility of epigenetic variation, of the inducibility of epigenetic variation, and the variable expressivity of epigenetic variations, which can be dependent on the number of generations of their own exposure, on how many generations a particular chromosome or part of chromosome had undergone sequentially oogenesis or spermatogenesis, on the genetic background. All these things we have to understand, to model, and once we will have good models, we will also be able to do good experiments, both in the lab and in populations. Of course, this doesn't mean that we should not do experiments now. We must have the ecological data, and this is being done. Ecological epigenetics is beginning to take off, especially in plants, but not only. So I think that what we need is a lot of population studies and a lot of modeling, because although people are doing modeling, individual people are doing modeling, and there are some very, very interesting models in the field already, it is not all put together. And certainly there is not one person who has taken it upon himself to systematize this theoretical framework, and we need this. Look, epigenetics is going to be relevant to everything. In the same way that genetics is relevant to everything. That you cannot, that after we have understood Mendelian genetics and genetics in general, and the role of DNA, for example, we understood that DNA variation is going to be relevant for everything. Every aspect of biology is going to be impacted. It will have an impact on everything. In the same way, epigenetics is going to have an impact on everything, on every aspect of biology. physiology and on our studies of morphology and on our studies of disease and of health and you name it, everything, because it is a fundamental part of what it means to be a biological creature. And if epigenetic inheritance is important, if the inheritance of epigenetic variations is important, then obviously it will impact, again, everything, health, disease, everything. So I think that the research that we are seeing today is convincing everybody, I think, about the importance of epigenetics and transgenerational epigenetic effects on medicine, on the well-being of people, and what exactly how profound it is. I mean, this is, work in progress, and but I think that if we will understand the theoretical possibilities, if we will be able to explore the theoretical possibilities in a systematic way, I think this will be a great help for cancer research, for epidemiological research, especially and for every aspect of medical research.
There you have it. It's everything. It's coming out and it's completely reshaping everything. But as you heard her in the very beginning, there's all this resistance because everyone was believing it went a certain way. We believed in the central dogma, the Wiseman barrier, natural selection. That's how it's done. Mutations occur, and that's what happened. When it's not mutations, it's epimutations. The genes that were already there just were turned off or turned on in the wrong fashion. And you can see all that mechanism working. And it's inheritable. Your environment changes these things. I receive from Google Scholar every new article that's written about epigenetics. I get 20 a day easily. I was just reading an article about stomach cancer being caused by a particular bacterium. I've done probably 2,000 stomach scopes called EGDs, and about 4 young ladies had stomach cancer. How could this be? A young person having stomach cancer. And I'll also test everybody for a particular bug called Helicobacter. All 4 of those ladies had Helicobacter. Sure enough, this article pops up yesterday that says Helicobacter changes the epigenes. It turned off the genes that prevented the cancer. I mean, can you imagine a bacterium was causing it and we didn't realize it? Because we were completely looking at the wrong stuff. We were thinking a mutation occurred and then that cancer sprung out. A mutation that changed the DNA sequence. But no, it's not. The DNA sequence didn't change. It's that the particular genes that kept the cancer away got turned off accidentally because of the bacterium. Totally different way of looking at things. So, I mean, autism is an epigenetic phenomenon. Aging is an epigenetic phenomenon. How our chromosomes shed their DNA. Illness after illness is popping up. I mean, the whole healthcare field really is epigenetics. And those 13 years of clinical research, all we were doing was pouring chemicals over things. We weren't doing anything to the DNA to cause it to actually change fundamentally because of our whole framework of everything, going all the way back to being taught evolution and how it has to work this way. And it was such a philosophical commitment to this approach that we completely stopped trying to falsify those things. And finally, we get lucky and figure it out. But it's really pissing off a lot of guys that are milking the cow and writing these articles. I mean, just the other day I was reading an article. They said, okay, the environment changes and certain birds don't chirp as much and therefore they're selected against. But guess what? Bird song is completely an epigenetic phenomenon. They didn't even talk about the epigenetics. It was easier to get a pass from a reviewer to say, well, this caused that. It's the typical Darwinian model, and I get a pass on my article. I get to publish or perish, basically. But they didn't go into the epigenes, which causes the bird song and other things. So they completely missed the point. Their article is wasted time, wasted money, basically. So we've discovered that genetics works in ways we never thought possible. There's a whole world of tags on the DNA and the RNA and all sorts of feedback loops involving all three components, the RNA, the DNA, and the protein. It's outside of Darwinism. It's outside of the Weismann barrier and the central dogma theory. This is an article that talks about the non-coding RNA. This is not actually coding directly for a protein, but it's telling the other protein what to do. And it admits that eukaryotes, those are cells that have a membrane to them, transcription is bidirectional. It doesn't go DNA, RNA, protein, but it goes the other direction. It's almost infinite potentiality and complexity, it says at the very bottom. It's blowing our minds, the sort of stuff that's coming out. What we wish to highlight here is the contrast between DNA that is mutated, right? The evolutionary model, DNA that's mutated, versus the chromatin, that's the protein around the DNA that is reshaped after environmental input. The mutation bears no functional relationship to environmental exposure. It's a logic of imprint, not of error. You catch that? It's how is the DNA imprinted to do things differently, not how it's mutated. That's basically saying, Darwinism is gone, basically, is what it's saying. But what about mutations? Mutations do happen, but are they random like that first film showed? This article just came out a month ago, basically, and it's talking about the very first parts of the DNA called the CPG islands. I think you may have remembered one of the first epigenetic clips. It was talking about CPG and the methyl groups and everything, turning on the gene or turning it off. Look what it says. The CPG islands are maintained by heterogeneous evolution, which is talking about epigenetics, regimes involving minimal selection. That's code word for no selection. Natural selection isn't occurring, guys, and yet that's supposed to be driving everything. Again, we have the CPG island, which is kind of like the piece of the film that gets the film into the spool so you can show it, and that starts it up, and then there's your gene, which makes a protein, and it depends on how many methyl groups is on this starter reel as to whether that gene is actually going to get turned on and get turned off. Well, guess what? According to that first film, a mutation has to occur to the gene, right, to make a change. But no, the majority of mutations are occurring on the promoter region. Has nothing to do with changing the gene. It just has to do with turning it on and off. And the majority of mutations actually occur on the DNA that has no function whatsoever. So how can it be naturally selected for?
You're saying a mutation, you're talking about, instead of a mutation of the actual gene itself, you're talking about mutations like as in when we see, for example, like a human born with like a monkey tail or, that type of weird thing on the exterior that we see?
No, we're talking about a mutation of, there's 4 letters to the DNA molecule, a changing of one of those letters to something else. And in fact, in this case, what it's really talking about is that there's a C and the G's over and over again. There's A, T, C, and G are the 4 different nucleotides. When in this case, we have a bunch of C's and G's, that line up, that starts this molecule that comes in, it grabs the DNA and it says, am I going to make a gene here? Well, here's all these lollipops stuck on there. I can't do it. So this gene is turned off. In the case of that bacteria I was telling you about, it turned off the good gene by its inflammation, or some food that we've eaten, or something else, or some influence is turned off. Our anger was visited upon the children to the 3rd, the 4th generation. Believe it or not, that sort of thing can turn off certain genes or turn them on in the wrong fashion. I mean, it's just mind-blowing, basically. But these little islands actually do mutate, and typically the C will get turned into a T, okay? But it doesn't do anything to the actual gene, it's just the promoter region. So the majority of mutations, which is the whole basis of the modern synthesis, it occurs in the parts of the DNA that doesn't do anything. So how can the concept of a mutation occurring and naturally select for it when it's a silent area that it mutates? The majority of mutations occur in a silent area. So basically, This is what the article is talking about. This elegant revolutionary rationalization for CPG islands is essentially neutral. Nothing happens. It does not assume any function for the CPGs. So if mutations do occur, it doesn't involve purifying selection. That's incredible to say that all mutations, the majority of them are occurring in the part of the DNA that don't do anything other than turning the gene on or off. So it's not changing the gene. It's just changing whether it's turned on or off. So basically, the CPG islands are being discovered to be regulated by epigenes, which are outside of natural selection. The CPG islands do not code for new protein. Rather, they serve as a promoter region for already existing proteins. We're not making new proteins here, guys. We're just maybe changing the way they're turned on and off with mutations. But CPG islands do nothing to change the nature of the gene. They only turn it on or off. Therefore, natural selection does not account for changes in non-coding portions of the DNA. It's a total fail in the neo-Darwinian model, basically. This is a gentleman by the name of Dennis Noble, who's a physiologist who had been saying for years that Darwinism wasn't making any sense. And he basically says the experimental results in epigenes and the related fields of biology show that the modern synthesis, neo-Darwinism, theory of evolution requires either extension or replacement. I mean, you can just imagine you're going to replace our theory, you know? I mean, millions of articles written by biologists around the world on natural selection, you're going to replace our theory. I mean, it's you know, it's making them very nervous, y'all. So, and then he goes on to say in an interview, the reason I think we're talking about replacement rather than extension are several. First, it's the exclusion of any form of acquired characteristic. You remember the Wiseman barrier? You know, we're not acquiring anything from our parents per se. And that's a central feature. You know, they beat poor Lamarck down, you know, conquered him and said, oh, Lamarck is wrong, all the high school texts. But this is in essence Lamarckian, which is completely different than the modern synthesis. So basically this is an article, Replace the Modern Synthesis, Neo-Darwinism, an interview with Dennis Noble. So here's Lamarck. I'll be back. Darwin, dang. Okay, so that's basically it, guys. I tried to condense it down in a way that we dealt with those basic 3 areas that kind of drove us in a certain way. We thought we understood what was going on, only to realize we completely missed the boat, and things are changing dramatically as we go. So we'll take some questions.
So you're basically saying what it boils down to, the reason that people are so resistant to it is because the old school thought is that because of all these mutations, we can add stuff in that makes us go from bacteria to multicellular to simple life forms to eventually we get bigger, we add tails, we add lungs, and all this kind of stuff. But the problem is that epigenetics is basically saying that you never add anything. The DNA is always there for lungs and stuff like that.
Exactly.
So if we believe that to be true, there has to be some sort of creator, there has to be some sort of like pre-design. Like you can't add genes that create a lung. The lung is always to be there.
That's right. Yeah, all of us here have 97% of the same active genes as a mouse, but we're not mouse, are we? I mean, it's, you know, it's how those genes are turned on and turned off. Say in this room here, we have 1x4s, and we have some glass, and we have studs and other things. Say there's a gene that made this, for instance, or a gene that made a smaller board. Okay, you can take all these components here and make a skyrise building, can't you? It's all the same stuff. You just got to be able to put it in all the same areas, in the right areas to make a totally new form. I mean, we only have about 150 different cell types in our bodies. How is it that they're so different? Again, as he was talking about, you're turning on certain genes, you're turning it off that build certain cell types. So that's why a mouse can basically be almost identical to us. It's also pretty good, by the way, that God made him that way, isn't it? So we can do studies, genetic studies on mice, kind of help us out at the same time. It's almost like he gave us this blessing of an identical, you know, mouse that can replicate and do things that's so different, so similar to us. A mouse is more similar to us than a hippopotamus or something like that. They're only about 60% the same, 80% the same DNA. A common fly has 50% of the same genes. but we're so vastly different because of the way the epigenes turn them on and turn them off. So now we're starting to understand it's not mutations that causes us to morph, like you've seen these morphing photos of going from one thing to the next. It's a very specific thing that's occurring. The genes were already there. They're just being turned on and being turned off. Well, that goes back to the original question, where did that gene come from to begin with? So it is a teleological look at things, because there was a famous philosopher of biology called Karl Popper, and he said, this is just a conundrum. How is it that DNA actually codes for something when it requires the product of its coding to start it to begin with? Well, that was a simple Karl Popper generalization. Now we're talking about, look at all those epigenetic tags that causes the DNA to express itself, that folds up that DNA that's 6 feet long into each of our cells. Millions of miles of information that are crunched down and opened up and done like this to spring out at the right time. It raises the bar of complexity, irreducible complexity basically, to a level that's just mind-blowing in essence.
Oh, no, I remember you saying that there was a lady biologist maybe in the 50s who was onto this but was kind of shut up, so we put way behind.
Yes, there was. Gosh, her name just, Barbara McCluskey. She was studying corn, and she noticed that genes were jumping in and jumping out of the DNA. And that just didn't make any sense according to the evolutionary model. They were saying, this lady's nuts, don't pay any attention to her. They'd go in their lab, they'd say either she's a genius or a nut. Well, she later won the Nobel Prize, so she wasn't a nut. She was saying this is going to blow everything up in 50 years because those genes, which they were talking about, are what our body is made up of. There's all these little jumping genes that pop in and pop out, and the epigenetics expresses them all in the right order. And 40% of our DNA is actually made up of pieces of genes that come from bacteria and viruses and other things. So this whole it's kind of going into a different area, but nevertheless, they do involve the epigenes and the epigenetic function. And so they called her pretty much a nut, but now she's, 50 years later, everyone was kind of focused this way. No, it has to be this way. It can't be the other way. They wouldn't even entertain it, basically. And then lo and behold, when that OnCode project came out just 2 years ago, and they suddenly said that much of our DNA is being expressed, they had biologists that were just violently aggravated. They were, a group of them wrote an article, and 7 of the people kept their name off the article. They didn't want to be known for criticizing. So one poor guy had to put his name out there, and there were 7 other guys that were silent, saying, "No, this can't be right. This ain't evolution. This ain't what we understand it." But the OnCode project information just keeps popping out. I get emails daily from the stuff that they're deriving from that OnCode data, which is just incredible. So this whole field is marching forward. It's leaving those guys behind. And what it's really doing also is it's scaring the heck out of them, because they had a good deal going. They could just write an article, just like that one I was telling you about the not getting enough food, and therefore natural selection. It made sense, right? It filled the dogma they would turn. But now they have to come out and say, no, wait a minute, what's going on with the epigenes? How is this affecting human illness? Diabetes is an epigenetic phenomenon. Weight is an epigenetic phenomenon. They discovered in Denmark during the World War II that if a woman was kind of really thin in her last trimester, her baby and the baby's baby would be thin. How could that be? Their DNA didn't change sequence. It was the epigenes changed. But if they ate too much in that last 3 months, probably like my mom did, then they're going to be overweight down the road. It's kind of like, dang, Mom, So it's now I'm telling this to some of my OB doctor friends. I said, look, tell your ladies, I don't want to see them for diabetes and high blood pressure and half a dozen things. Tell them to take it easy just for 3 months. Those last 3 months, take it easy. And their kids, isn't that amazing? I mean, here we are trying to do exercise and take pills and buy Nordic tracks when all we had to do is catch some others that last 3 months and tell them to take it easy. So, so that's, again, we're getting back to the sins of the father, to the third and the fourth generation. There it was all along,
Well, first thing I think you need, I mean, this is great. I think you need to give Richard Dawkins a call, because he's obviously not heard anything. He still thinks we're all the product of innumerable genetic mutations over long periods of time. But the evolutionary community is, there is no more politically correct group than them.
Yeah.
And they're not going to take this lying down. I mean, they're, because this, as this fellow said, this smells a lot like it's guided. It smells like there's something behind it other than randomness. So I'm just thinking about how it's going to play out. How is the evolutionary community, the Darwinians, when you think that way, genetic mutation plus natural selection, how are they going to fold this knowledge into their beliefs?
Well, the way they're doing it, and you heard Eva Jablonka said she was still throwing the word evolution around. Evolution, you like the color blue, it's evolution, right? I like the color red, I mean, they just throw it around that can do everything, basically. But they still want to believe in some form of change, and it is change. The change, though, is very teleologically set, that the information is already in there, and it changes one way or another depending on our environment and the things that we do. They're coming up with a concept of common extended synthesis. But the problem is they're trying to hold on to their Darwinian thoughts. And to hold on to their Darwinian thoughts, they have to hold on to the Wiseman barrier. They have to hold on to the central dogma. All these things which are completely wrong. And, natural selection, which is, mutations causing real change when mutations aren't causing change. They're hurting at first, okay? They don't want to have to do this stuff. But even P.Z. Myers, who's one of the biggest evolutionary dogma guys, hurdling stuff all day long on his website called the Ferengula, I believe, and he admits that a third of the students coming into biology are going to study epigenetics. That's where the money's going. $20 billion from industry has been committed to this. I mean, you saw what $250 million did to discover the ENCODE project. Google's throwing in $20 million just 3 months ago. Everybody realizes this is where it's at, so they're just gonna have to, they're gonna, after they start doing this stuff, they're just gonna kind of give up the old theories. They're just gonna, fade away, I guess, Does
this system create a new species?
No, that's the problem, isn't it? No, it's taking an existing gene and turning it on and turning it off to make a different cell type. But it had to be there. Where did it come from to begin? That's a question that's thrown it back to those guys. They're going to have to, they can't explain natural selection causing epigenetics. There's no connection to it because you have to have a mutation that causes it. And as we saw in that last study, the mutations are causing changes in the neutral parts in front of the DNA, the gene. Most of the mutations are occurring in those CPG island areas. Well, how can we then change a gene, for instance, to cause a change in the CPG islands? We can, That's it. I mean, and that's why I love that verse. God, it's the glory of God to kind of keep things a mystery. He kept it a mystery. He let them keep putting it out there, keep waving their arms, keep pumping Darwinism up. Every time it almost fails, they keep pumping it up. They can't pump it up much any longer as this stuff comes out. They're just simply going to fade away, really. And the articles 2 months ago, when I do searches on natural selection in Google Scholar, the articles on epigenetics surpassed the term natural selection, which means that these guys are milking the cow as fast as they can, but they can't keep milking it too much longer because they're going to realize they have to go back in there and explain all the epigenetic tags along with the natural selection, and it falls apart. So
from a reasonably point of view, we would say that epigenetics cannot explain the progression of life or the history of life that we see on the earth without direct intervention?
Well, that's just it. Now that's where it really gets interesting. You know, could it be that God started it all at the very beginning with all this mechanism that's in there, and then it does change things? I don't know. I was talking to Hugh Ross about this, and he was actually made the statement that I was kind of talking about, that it may be that the epigenetics is causing new life forms to pop out. There was an interesting article that came out where they took flatworms. You know, worms, you know, you can break a worm in half and it can regenerate itself. So it had this regenerative capability. And they put it in a particular chemical bath which shut down the cell's communications with other cells around it. The flatworm expressed a head that was of a different species, a completely different species. In other words, the DNA was in there to actually cause different species. And once it went away, it popped back to its original species. So it could be that we have a multi-potentiated DNA to cause different things.
And there's a video, somebody may have seen it, but it's going around on Facebook right now, of where you put a banana together with a kiwi and it turns into a banana kiwi, literally. Yeah, I mean, literally, you just cut it and put it together.
What you're talking about is grafting, okay? About 4 months ago, they came out and they were showing that when one plant grafts with another, epigenetic RNA is going from one plant to the other and turning off certain genes and turning on certain genes. How in the world could natural selection have occurred in that fashion? Think about it. Natural selection takes millions of years or whatever of change, small change, but a plant breaking and flying into another plant, maybe during a tornado, how's it going to suddenly live? It'd have to, you know, look at all the things that would have to occur to accommodate it. This means that the DNA was actually able to accommodate epigenetically. This might explain why certainly at certain times when after certain major events, like maybe the Cambrian explosion, that certainly whole new animals started popping out. You know, after oxygen started rising in the Earth's atmosphere. We don't know. I mean, this thing could really go a lot of different ways. So it may be that God was such an incredible designer that he put it all out there and it's just kind of finding its way. This is not theistic evolution, by the way. It's taking genes that were already there to use them in different fashions. Theistic evolution assumes that new genes develop by mutations, but again, it's not that at all.
But I could see theistic evolution saying that works perfectly fine with us.
Yeah.
It's a natural way of things occurring. I'm curious, ask Dr. Ross's Facebook page, because I would assume he had a different meaning behind what he expressed, since he believes God creates individual species of genuses and not any kind of evolving from one to the next.
We really just don't know yet. I mean, this really
that
is.
would be something to discuss with Dr.
Yeah,
Vaughn.
sure. And in fact, I did talk with him a bit. He's really kind of focused on certain other areas right now, and they're trying to get somebody with their group that focuses exclusively on epigenetics, because it could mean a lot of different things. I mean, if you think about it, they were kind of stuck in a gene-centric model. You know, the genes were changing gradually to cause this new thing to happen, when it could have been that the genes were already around, they just started getting used differently.
Because I know different types of creatures have different numbers of genes. So somewhere along the line, genes have to be inputted for certain types of creatures to exist, and then the epigene then works on those existing genes. So if those genes don't exist in this one creature, but they exist in the next creature, one creature can't epigenetically change into the next one because the genes are totally different.
Well, that's true, but think of the fact that we have 50% of the same genes as a fly does. It's our epigenes which use those genes differently. Or we have 97% the same as a mouse. It's a totally different way of looking at things. Now we're understanding that the DNA are the little specific genes, and not only just the genes, but by the way, they were doing a study in the fruit fly and discovered that one gene can be used in 30,000 different ways. So, I mean, we're just talking about our minds just want to go like that guy, pow, you know, it's just every time something new. However, the information that causes one gene, how do we get for that information? The mathematics is incredibly difficult. I I mean, there's 4 different letters, and each time you add another letter, you add to the power of 4. So, okay, you got 1 letter, A, T, C, or G. 2, you take 4 times 4, and that's 1 out of 16. Add another 4 in there. So 1 to 100 to, was it 64? Times 4 is 256. That's just 4 DNA molecules, 1 out of 204. By the time you go out 20,000 genes or something, you're talking about 4 to the power, of 1,000,000 or something. It's just, the mathematics are just astounding. They can't figure out how that happens. They were saying, well, maybe mutations occurred over millions of years to cause this. And that was the argument from ignorance. We can't know it, we just know, we know evolution did it, therefore it must have, right? We're here, evolution did it, right? That's the kind of the circular taunterism of a circular argument. You can't argue against that. You can't, well, guess what? All these different organisms have 20% different DNA that recently came along, okay? I read this one article, this was just from a few years ago, called Darwinian Alchemy. It was hilarious, basically. Sad, but hilarious. At the end of the article, it said, imagine a new gene coming into existence and then getting more complex. That was its mechanism, imagine. I posted that in a large blog site that had 3,000 evolutionists, and I put John Lennon, Imagine, and playing, and they were like, oh, I said, that's what it's saying. It's saying, imagine a gene coming into existence and getting more complex. And one guy finally said, thanks for posting it, but would you stop posting this article? Because, their whole phase system is not even able to be reasoned out, how a new gene even comes into existence. That was it. I mean, imagine the process occurring and then becoming more complex. If y'all are satisfied with that, that's great, but that's not an answer, really. Mathematically, it doesn't explain anything. But now you add to this level of complexity, epigenetics, you can't even imagine it. Like that article said, it's almost beyond complexity, the different possibilities that can occur. That's why we can't formalize the language of the DNA. We can't fully put it to computer language because it's self-programming. It's able to change according to the environment. But it's also very important that our environment and the way we do things can affect our children. So it's very important for women not to be under stress, for instance, when they're pregnant, or when that baby's born, to have 2 parents. They've shown epigenetically, if you don't have a male and a female, that child is going to have problems when it grows up because of the way it affects the epigenes, the behavior of that child. I mean, it speaks to, the concept of a married couple, having both partners. And then you look at families where there's mothers without fathers and those children having problems. Now we're starting to understand a lot of that's epigenetic problems. Their ability to handle anger, interact with other people, depression, other things are affected by that. They did a study on women that survived the Rwandan crisis, right? You remember the Hutsis, and the Tutsis started killing one another, like 800,000 people killed. The women that were pregnant during that whole thing, their babies were born with post-traumatic shock. And guess what? Those babies and great-grandkids are going to be born with that, believe it or not. It's insane, but it explains how it is that certain cultures, maybe they have some sort of religion that causes a lot of frustration or anger or angst, that they can be full of anger generation after generation. You can bring them over here and they still want to bomb things, like the Boston bombers. They had everything they needed, but they were still full of angst and things from the epigenes being screwed up. The sins of the father to the 3rd to the 4th generation. It actually explains things like original sin, because when Adam and Eve ate of the tree, basically, they lost fellowship. They didn't die immediately, did they? Guess what caused them to die? Being put out of the garden and not having access to the tree of life. Well, something was going on with that tree of life that would have kept their epigenes all working correctly, but they were taken away from that. But what happened to Adam, for instance, when he sinned? What was the first thing that happened? he was told to cover himself. Tremendous shame. You know, Satan said, you're worthless, you're no good. You're gonna have to cover yourself up because you'll never be able to be close to God again. God's told him, he said, who said you were naked? Eve didn't say anything. It was Adam who was full with the angst of what he did. He understood what he had done. He was not deceived. He knew that he was choosing for her because he wanted to die with her necessarily. Okay? I mean, in a way, it's kind of very romantic, really. He wasn't going to leave her thrown out on her own. He said, I'm going to go with her, bite the apple, or whatever it was. But he knew, and he was separated. He had stress, and that stress is passed on generation after generation. The original sin is full of stress and angst. A baby is born, it cries. It's, you know, if it could kill you to get something, it would. As a baby, it has no, you know, it's a totally, you know, emotional being, and all those things are related to the epigenes of the sinful nature. That's why we sin, basically. And they've actually done studies on this to show that stress and everything does go through the male genes in this certain particular fashion. Females can do things as well, but just like that Bible verse, the hatred to the third and the fourth generation through the father, and they're discovering this is actually an epigenetic phenomenon. So now we actually have an understanding from a physical aspect what makes us have the old nature in us. And that's why we're going to have a new body one day to get rid of those bad epigenes. So,
what does this do on drug therapy versus natural therapy? When you think of, you just said they're dumping chemicals in people and they're going, whoa.
Okay. Mike here works with my clinic, and I was talking about the whole new field of epigenetics. We have epigopharmacogenetics. That's a new word, basically. Okay, we've had all these drugs out there for years. Now we're going to have to go back and see what they're causing to our epigenes. Okay, it's like, and that doesn't happen in 7 years like they're doing now. They're going to have to go back and look to see, are we changing the epigenetics to screw things up? This is going to change everything for the drug industry. For one thing, they can say, well, we get to have a longer patent because we're going to have to study this stuff for 100 years, basically, before we put something else. But these medicines do affect us epigenetically. There was a blood pressure pill that I used to, it was an old blood pressure pill that would occasionally cause rheumatoid arthritis. And you're like, what in the world? How in the world was this doing it? Because it was affecting the epigenes that affected the inflammation and everything. So we know that that drug has an epigenetic effect. There's other drugs that can have epigenetic effects to cause remodeling of the brain and things like that, so that we become more plastic again. So it works on conditions like manic depression and things like that. So it's like a whole new field is opening up. We're going to have to study the epigenetic aspects of the drugs, and until we do, we don't know whether we're really doing good or bad, really, other than certain mortality studies. That is always interesting. I can remember when I was a young doctor starting off that there would always be this old doctor who raised his hand in the back and said, what is this doing to our mortality? Does it actually affect mortality? And the drug rep would always bow his head because he couldn't really say it was affecting the mortality. You know, so what's the use of your drug? It was a concluding remark that the old crusty doctor would say. Now I'm starting to become one of those old crusty doctors where I really question, what are you really doing here? Are you doing any good at all? And you know, a lot of the times they have to admit that, well, we're really not necessarily changing mortality. So, so there's a lot of things that we have to study, a different approach to medicine completely, really. So
would you say our DNA already knows, I guess, every virus, bacteria that's already out there? This is like,
well, yeah, that's an interesting thing.
It's happening just because it's the gene actually turning on or off.
Think about your computer, and a virus comes flying through. What does it do? It takes that piece and it walls it off in a certain area. In our DNA, it does the same thing, but it says, hey, look, I know that this is bad. I'm going to use this as a constant reference, so when a new piece comes in, I'll compare it to that old piece. So in essence, it's using bits of DNA, and we have all kinds of DNA from other individuals to know what is good for us and what is bad. We have to carry a copy around. They were calling that junk DNA, but it has a great function, actually. It keeps us from dying from all these viruses and things. I mean, all our DNA is actually kind of overlapping in a lot of fashions. So, yeah. So,
with like the innate and adaptive immune system, is stool.
Well, that's an interesting thing, that the immune system change. Are you talking about like antibiotics and things like that? Well, the evolutionists always say, oh, isn't this interesting? You know, the fit survive and then they go on to have children. Well, how did they survive? It's because they already had that DNA in them to make a change. Where did that come from? It didn't occur instantly. If you wait for natural selection, you're wiped out with the first salvo of antibiotics. No, that DNA was in there. Now, it's true that, like, you know, that they are, quote, fit, they survived, their kids did that. But as it turns out, many of those mechanisms are actually bad mechanisms that cause bad nutrition and everything else. So once that cell gets back into a natural population, it dies off. And not only that, but it doesn't ever get full memory to go to the past. Like staph, for instance, is now forgetting how to handle sulfa drugs. One of the first antibiotics we ever had. It's learned how to handle the new stuff, but it's forgotten the old stuff. Yeah, so it's a completely different way of looking at things, basically. So, yeah.
Just out of curiosity, do you think the mechanism you're describing explains the apparent physical differences between the various races?
Yeah, it does. Yeah, I mean, because one race can go out and have a different environment, and it can change the way they are. They can adapt rapidly. So, but the problem with adaptation is the term automatically carries a lot of evolutionary baggage. What they're doing is they're coming back out and renaming it. They're calling it plasticity, heritable plasticity, so as to get it away from the old evolutionary terms. Okay, it is a form of adaptation, but it's immediate adaptation. That's why when different species go out there and get around different environmental changes, they can change dramatically. Okay, look at Darwin's finches, for instance, the darling of evolution, right? They started studying the parts of the DNA that has to do with beaks. You know, what makes the beaks different? And guess what? It's epigenetic tags that make those beaks different. It was all epigenetic. It wasn't natural selection. And they change by the season. When it's a particular dry season, their beaks change differently. And when it's wet, they change back because the epigenetic tags turn on certain proteins and turn off other ones. It wasn't natural selection at all. It was epigenetics.
Can this happen in an individual's lifetime? So in other words, I'm always replicating, the cells replicating the DNA, the epigenetics change.
Yeah.
And I'm different from 10 years ago to...
Yeah, actually it does. It does. The epigenes are involved with cellular aging, for instance. Now, it's a mechanism that, for whatever reason, we've lost our ability to keep our soma going, if you will. And it has to deal a lot with the epigenes on the ends of the chromosomes called the telomeres. And it's kind of like our chromosomes just kind of shed DNA at a certain point, just wears out. If we could fix that epigene phenomenon, it could be a matter of diet. When God comes back, he may just kind of walk into a lab and say, here, add some lime juice with this and a touch of vodka and that, and you'll live forever. And we're like, oh, kind of like face palm, I mean, it could be something simple as that, I mean, you think about it, supposing when he comes back, we're going to have this millennium where we're not going to age, all this, and yet we're still going to be sinful. It actually kind of answers a question. Everyone says, well, if I lived in the garden, I would have done differently. Well, in the millennium, there's going to be a countless number of people who don't have to die. They're going to have perfect government. You're not going to have situations going on with whether it's Ted Cruz or Donald Trump or Hillary Clinton or whatever. We're going to have a perfect government. And at the end of it all, what happens? It says that after Satan is released from his bondage for 1,000 years, that there's going to be so many people, they're going to be like the sands of the earth, they're going to revolt against God. Even under perfect environment, they're going to still revolt against him. It just goes to show that even with perfect epigenetics being reestablished by the knowledge of God, our ultimate solution is a resurrection body, a new body. We have to finally get rid of the old man ultimately, because even if we could live 1,000 years, we're still going to have these sinful tendencies. So, very interesting.